Hello everyone. In the last session, we talked about protein structures, and in the previous sessions also we talked about the importance of protein structure prediction and also protein function prediction. Why is that? Why is it important? Let's remind ourselves, because assume that you know about the known protein structures, you know about the known protein functions, and now if we are dealing with a disease or disorder, that we know that in this disorder we have lack of production of a specific protein, then in that case, if you can produce a new or maybe another protein which is known and predicted that if the structure is very similar to the protein that in that body, or in the, for example, we are talking about the patients who are suffering from lack of that protein. So if we can discover such a thing, such a protein, or a replacement for that protein, then we can produce it more in the body using gene therapy, using drug therapy in order to compensate it. So this is a big deal. That's why it's very important to predict function or structure of protein. And specifically now we are talking about the structure, because if you find out about the structure, most probably if they share the same structure, two different proteins, most probably they have the same function too. We already talked about secondary structure of proteins. We mentioned about alpha helix and also beta strands, and there's another component which is important, coil. So basically we can mention about 3 components instead of 2 in secondary structure if we consider coils as well. So assume that you have a primary structure. Primary structure, it means that the sequence of amino acid that we talked about before, just starting from the first position going to the last one. Okay, so we talked about it before, and it's possible to represent it by secondary structure in order to convert everything into just these 3 components of the secondary structures. So now everything will be converted to C, E, or H, coil, alpha helix, or beta strands. So now we are dealing with 3 letters only. This is even now easier than DNA sequence, right? Because for DNA we had 4 nucleotides, now we have 3 ones. So it makes our job easier, and also it makes it possible for us to understand about some regions. You see, now we can compare regions together, like the local alignment that we talked about before. Now it works very good for secondary structure prediction as well, because we are just focusing on a specific part of the sequence and try to compare it with other sequences. And yes, local alignment could work very well in that scenario. But generally, what methods, what techniques do we have for protein structure prediction? Mainly, we can categorize them in these 3 methods. The first one is ab initio technique, which is starting everything from the scratch. And so when you want to do that, it means that you should check everything, and it's computationally expensive. But the good thing about this technique is that this is flexible. You can apply it for different types of proteins that we are looking for its prediction. And the other one is homology methods, and this one is comparison of sequence with another sequences. And this one we already talked about, because homology, we talked about it in details, that what do we mean by that. And if you find out about the common functions of 2 different genes, and of course, when we talk about the function of gene, it means that a gene produces, synthesizes a protein, and that protein is responsible for a specific function when I say about that. But the thing is that we already talked about how we can compare sequences together. We talked about alignment, pairwise alignment, multiple alignment. So this is pretty much the same thing. And for protein threading part, this is new for us. We never done it before. It's like comparing a sequence with a structure. It means that in the protein data bank, for example, we have some known structures for proteins, the 3D format, for example, and they are limited. There are not infinite number of them, and they are not 100% accurate, but we have a data bank for that. And you can compare your sequence with that, and we talked about that in details. We can also categorize amino acids into different components. Like, for example, you can discuss about them in terms of hydrophobic or polar factors. So hydrophobic and polar factors could be also considered as one of the important factors in order to categorize amino acids. And it means that if you consider them, now transform everything into this sequence of series of P and H. Now we have only 2 letters. You see, it's even easier to deal with such sequences now. It's like 0 and 1s, just binary. So it's also another way to deal with them, because when we know about these characteristics of amino acids, then it's also easier for us to predict its function. For example, if we have too many hydrophobic ones together, then what's the function? Is it a good thing or bad thing that we have hydrophobic ones together or polar ones together? For them, you should know a little bit chemistry. That's why we don't go that much into details, as you know, to different sciences, but it's nice to have background about these things, because it's helpful to know what we are talking about at the end. And for amino acids, if you search on internet, you will see exactly which amino acids, for example, has which characteristics. So it's not possible, for example, one day this is polar, the other day is not polar. It's always the same because it has the same atoms, the same binding. So the characteristic is fixed, and you can easily transform everything in such a sequence. But the thing is that when I talk about, for example, this position and this position, when we are talking about primary and secondary structure, the problem is that we don't see exactly how close are these two, because we are seeing it as a line, as a position. Okay, as a position, we talked about alignment and we saw how we can compare them. But when we talk about proteins, we are dealing with 3D structure. It means that maybe, for example, from this position to this position, we have a fold, and if we are considering as a strand, and inside the protein, if you look at it, it looks something like this. So it means that from this place, maybe we will have a bending, and we will have such a structure, for example, from this P to another one. So these two actually are pretty close, you see, because we thought on the primary or secondary structure that they should be far away, but this is not the case in the 3D structure. So that's why it's very important to also consider 3D ones. And also there is another format which shows the 2D, and still it's very good to get ideas what's going on in terms of their combination. Now, look at this scoring lattice models. In these ones, we can consider them as 2D and 3Ds. And you see, when you show them in such a format, we get better idea that which amino acids are actually closer to each other. In primary structure, we couldn't see that. So these two actually are close, but we didn't know that. You can even consider it in terms of 3D, which is much meaningful. Now it means that you will have 3 variables, and you can show the position of each one of these amino acids. And if we can do that, then it means that you can find out about their exact distance. And when you find it, you get better idea of what's going on inside the protein structure. And we will talk about it in details also next week, because I will introduce the algorithms related to that, how we can calculate it, and how we can predict the protein structure. This week mostly I try to focus on introducing the background related to it and what's the goal here, what are the components, because when we talk about this specific model, then you should know exactly what we mean by that, because directly we go to the algorithm next week. So for the alignments, you remember, for example, for matches, for example, gaps, we had different scores. For example, for matches, we usually give them positive values. For gaps, we usually give penalties. For the scoring lattice models, we do the same. It means that is it good that, for example, 2P is close to each other, 2 edges are close to each other or not, or far away from each other or not? These things also we can give positive or negative values to them, or higher or lower value to them. So we will talk about them also in detail. That's why it's important to see, for example, in 2D or 3D formats, that what's the relation between them. In 3D, as I said, this is the best way, because we can exactly show the position of each one of the amino acids. So one by one, we calculate all of them. For example, for one specific amino acid, we say, for example, for X, we have this value, for Y, this value, and Z, this value. And also, we can show their primary structure position beside them as well. So how we can construct such matrices, we will talk about them for sure. What are the main advantages of protein trading models that we talked about, which are comparing sequences with proteins? I mean, the structure of proteins which we know. So structures can adopt wide range of mutations. We talked about different mutations before, that sometimes we have a point mutation and it doesn't affect the protein structure. It will give you exactly the same amino acid, for example. Or maybe we have a region for our mutation, and still we will reach the same structure of the protein, or similar, pretty similar structure of the protein. So such protein trading models could be a good idea, because we are comparing structures with sequences, not sequence with with another sequence. Because when you do that and you penalize the alignment, for example, for gaps and everything, then it may gives us wrong ideas about the structure of the protein. It doesn't give us any information about that. That's why protein trading models could be very useful. And also another advantage we could mention is about number of known protein structures are limited. As I said, in the protein database, if you look at it, you will see limited number of proteins. So it's much easier to compare something with limited number of structures. For example, instead of having various sequences, we already talked about the probabilities that, for example, if we have exactly 3 matches in the same sequence, what's the probability? Then if you have long length of sequences, then we have so many options, so many possibilities. And that's why it makes it very hard for us to compare one sequence to another to get idea about the uh, protein structure, for example. They are useful. It's not like we should just throw it away because they are very useful. That's why we talked about alignments before, and we introduced you different algorithms. Also, we mentioned that they are heuristic. It means that the optimal answer that you will find is not 100%. Sometimes you eliminate many options in order to reach the optimal answer and guess, for example, with a specific probability that yes, maybe this is the optimal answer that I'm looking for, and they are working. And that's why we cannot say, for example, comparing sequence with another sequence is useless, even for protein structure prediction. Now, for scoring alignment in protein threading, we have the same scoring alignment, also measurements in protein threading models as well. It's not just for comparing sequences together. So what we do here, given a sequence and a fold, what do I mean by fold? It means that a specific protein structure that you can find also in the protein data bank, or we can also call it template. We can compute the optimal scoring alignment between the sequence and the fold. So again, for comparing sequences with different folds, we can now find scoring alignment, and we will talk about it also in details about the algorithms that we can use in order to find them. And then based on the scoring alignment and checking a new sequence with known folds, so known folds, where do we know them? From the protein data banks. So they are not 100% accurate, as I mentioned, but as much as possible, they try to make it clear by using different methods like X-ray to get the idea, for example, where is the position of the atoms. Because it's a 3D structure, then they try to predict where is X, where is Y, where is Z for each atom inside the protein. So different methods in order to find them. And there were big projects for them, obviously. But fortunately, we can access them through that data bank easily. Now, assume that you will find by a specific algorithm about the scoring alignment. Then we need to calculate test statistics and the corresponding p-values related to them. We talked about it in the statistical significance lecture. We don't go through that much details because in another course for statistics, we are talking about it. So we are evaluating you about finding test statistic, p-values, and everything inside the statistic course. So for this one, we are not going through that one in details, but you should know about the goal anyway. That at the end, everything is in terms of testing a hypothesis, because we are claiming something. Then based on that, we want to see whether it's true or not, whether the structure of the protein is similar to the other protein or not. Is the function similar to the other protein or not? And based on this hypothesis test, we can find it. And all of them are based on test statistics and p-values. That is, everything is based on probabilities. Everything is based on confidence level. It means that nothing is 100% again. For example, we say 95% I'm sure that this protein will have this structure or have similar structure to this protein or have this function. So this is the, I think, perspective that you need. This is the vision that you need in order to go through the examples that I will give you, the algorithms that I will mention starting from next week, that how we can deal with protein structure predictions. Okay, guys, if you have any questions, please let me know. Otherwise, on Wednesday, we will talk about that weekly assignment related to phylogenetic. And also next week, we will start the algorithms related to the discussions that we had today and in the previous lecture.